Human survival requires converting chemically complex raw food materials into simple, absorbable nutrients. These nutrients provide the baseline molecular fuel for cellular energy, continuous growth, and tissue repair. This conversion occurs within the alimentary canal. This is a continuous 6-meter-long tube-like structure spanning the human torso, extending from the oral cavity to the anus. The operation of this canal relies on 3 foundational processes: ingestion, propulsion, and mechanical digestion. Mechanical digestion physically fragments the ingested food. By breaking large solids into smaller pieces, the body drastically increases the total surface area of the food mass, preparing it for the chemical breakdown that follows. To move this material through the system, the body uses propulsion. The primary mechanism is peristalsis, involuntary alternating waves of muscle contraction and relaxation within the organ walls that force the mass forward. The pathway begins at ingestion in the oral cavity, passing through the pharynx and down the esophagus. The mass then drops into the stomach, is propelled through the coiled small intestine, the duodenum, jejunum, and ileum, before its final transit through the large intestine. This 6-meter pipeline provides the physical space and mechanical force required to crush and transport food. However, mechanical action alone cannot extract the distinct molecular fuels the body requires to function. To extract that fuel, the alimentary canal relies on accessory organs. These are supporting glandular structures that sit outside the digestive tract, but secrete essential fluids and enzymes directly into it. The largest gland in the human body is the liver. Positioned just under the diaphragm, its primary digestive function is the continuous production of bile. Tucked beneath the liver is the gallbladder. This dark green sac acts as a storage and concentration reservoir, releasing the liver's bile only when active digestion requires it. Lying retroperitoneally behind the stomach is the pancreas. This lobular gland serves 2 entirely different systems, possessing both endocrine and exocrine functions. Its endocrine role involves producing hormones like insulin and glucagon to regulate blood sugar. Its exocrine role, which is vital to the gastrointestinal tract, involves using numerous acinar cells to secrete massive volumes of digestive enzymes. These accessory organs engineer the required chemical environments. Without them acting as external chemical factories, food would travel the entire 6-meter pipeline and exit the body completely unabsorbed. Chemical digestion begins the moment food enters the mouth. The parotid, submandibular, and sublingual glands secrete between 800 and 1,500 milliliters of saliva daily. This saliva contains mucin, which lubricates the food bolus, and an alpha amylase enzyme called ptyalin, which immediately initiates the chemical breakdown of starches. This graph illustrates the drastic environmental shift as food drops into the stomach. Submerged in gastric juice, the pH plummets to a highly acidic 1.5 to 3.0 range. This extreme hydrochloric acid environment denatures complex proteins, physically forcing their tightly folded structures to unravel. Once the proteins are unraveled, the gastric enzyme pepsin utilizes this highly acidic environment to sever them into smaller peptide chains. Alongside these digestive acids and enzymes, gastric juice also secretes intrinsic factor. While playing no direct role in breaking down food, intrinsic factor is absolutely critical for the small intestine to absorb vitamin B12 later in the pipeline. Crucially, the stomach does not absorb nutrients. Its chemical objective is to dismantle tough protein architectures and liquefy the ingested food into a highly acidic paste known as chyme. A chemical crisis occurs as this highly acidic chyme exits the stomach and enters the first section of the small intestine, the duodenum. The intestinal mucosa cannot withstand extreme acid. Pancreatic juice rescues the intestinal lining. Secreted from the exocrine pancreas, this juice floods the duodenum with heavily alkaline bicarbonate ions. This neutralization process raises the internal pH to between 6 and 8. This creates the optimal environment for specialized pancreatic enzymes to deploy and finalize digestion. First, pancreatic amylase aggressively targets carbohydrates, converting remaining starches and complex sugars into simple, absorbable monosaccharides. Second, the enzymes trypsin and chymotrypsin target the denatured proteins, severing the remaining peptide fragments into individual amino acids. Digesting dietary fats, or lipids, presents a unique mechanical problem. Because fats are hydrophobic, they clump together into large globules that repel the water-based enzymes trying to break them down. Bile solves this problem. As noted in this text, bile acts as a biological detergent to reduce surface tension and break apart large fat clumps. This process is called emulsification. By forcing the fat to collapse into tiny droplets, bile drastically increases the total surface area available for enzymatic attack. Once emulsified, the enzyme pancreatic lipase can successfully interface with the lipid droplets, converting the triglycerides into absorbable fatty acids and glycerol. Through this precise choreography of shifting pH levels and targeted enzyme deployment, the duodenum successfully isolates the body's three ultimate cellular fuels. First, the monosaccharides, next, amino acids, and finally, fatty acids. The remaining 5.5 meters of the small intestine, the jejunum and the ileum, are responsible for the vast majority of nutrient absorption. The isolated monosaccharides and amino acids are absorbed directly through the intestinal mucosa, entering the bloodstream for systemic distribution to the body's cells. Fatty acids utilize a different biological pathway. They absorb primarily into the lymphatic vessels before eventually joining the venous blood supply. By the time the remaining material reaches the large intestine, all useful macronutrients have been stripped away. What is left is a liquid mixture of undigested fluids and non-starch polysaccharides. The colon's primary job is to recover water and electrolytes from this remaining waste. As water is reabsorbed, the indigestible residues solidify and compact into feces for defecation. Nutrient absorption is the physiological finish line. It is the precise moment food officially transitions from existing in a tube outside the body's internal environment to becoming a physical part of the blood supply. Clinical pathology begins when this physiological pipeline breaks down. Accurate nursing assessments rely heavily on identifying exactly where and how a mechanical or chemical failure has occurred. Peptic ulcers represent a failure of the protective mucus barrier in the stomach or duodenum. Without this buffer, highly acidic gastric juice directly erodes the underlying tissue wall. Diarrhea is a motility failure. When transit through the large intestine is too fast, the colon does not have the necessary time to absorb water, resulting in severe fluid loss. Conversely, constipation occurs when transit is abnormally slow. The colon absorbs too much water from the waste mass, resulting in hard, impacted feces. Jaundice acts as a systemic indicator of a liver or biliary failure. When these organs lose the ability to process and excrete bilirubin, the pigment builds up in the blood, causing a visible yellowing of the skin and eyes. These visible patient symptoms are direct, logical consequences of chemical or mechanical breakdowns at specific geographical points in the GI tract. Nurses utilize this physiological map to guide patient care. Baseline assessments include calculating body mass index, auscultating for bowel sounds to confirm peristalsis, and tracking strict fluid balances. Diet management requires anticipating surgical impacts. This dictates maintaining a strict NPO status before procedures, or verifying the patient receives adequate complete proteins postoperatively to support tissue repair. In severe cases of pathology, such as colorectal cancer, surgeons must completely remove portions of the colon or rectum. This requires the surgical creation of a stoma, a colostomy or ileostomy. The surgeon permanently diverts the end of the healthy bowel through a small opening in the abdominal wall. Managing this alteration is a highly specialized skill. A wound ostomy continence nurse selects the precise stoma site, educates the patient to prevent appliance leaks, and meticulously protects the surrounding peristomal skin. Understanding the exact flow and chemistry of the digestive tract empowers nurses. It allows them to anticipate nutritional deficits, effectively manage surgical interventions, and help patients adapt to permanent physical modifications. To review, the alimentary canal provides the physical pathway. The accessory organs deploy the chemical tools. The end result is the extraction of simple sugars, amino acids, and fatty acids. Take a moment to test your retention of this material against these 5 core clinical competencies. Pause the video here to review these questions. Ensuring you can accurately answer them will verify your foundational understanding of the gastrointestinal system.